Well, welcome everyone. People are beginning to look more familiar as we get to the later segments. I've seen a lot of you coming and going through the corridors and walkways. I'm Joe Palca. I'm moderating this session. This is about vaccines, and we have, about as good a panel on vaccines as you could ask for. To my immediate left is Tony Fauci. He's the director of the National Institute of Allergy and Infectious Diseases. Next to him is Filip Dubovsky, who's the vice president for clinical development for vaccines at MedImmune. Next to him is Gary Nabel. Have I got that right? Yes. Hi, Gary. Director of the Vaccine Research Center at the aforementioned National Institute of Allergy and Infectious Diseases. And next to him is Peter Agre, who is the director of the Malaria Research Institute at the Bloomberg School for Public Health at the Johns Hopkins University in Baltimore. And the format is going to be informal. I've asked each of the speakers to say a few opening remarks or not, and then we'll talk and entertain your questions. So I'll start with, and I'm going to, if it's all right with everyone, I'm just going to go down the row this way. And so that'll mean we start with Tony. 5 minutes, yeah?
Good. Okay, thank you, Joe, and welcome, everyone. I'm going to just discuss a couple of concepts to sort of set the stage for what we hope would be an active and lively discussion after. So I want to make 4 points during the few minutes that I have. First of all, I'm sure most people in this room know the history of vaccines and vaccinology is really most extraordinary. When you think of public health measures, I think it's unarguably the most effective public health tools that we've developed when you think in terms of the numbers of lives that are saved per year by the implementation of programs of vaccine preventable disease. The estimate conservatively is about 2 million lives per year that are saved. For every $1 of investment in vaccine development, you have about $18 to $19 saved by society, which comes to about $40 billion a year by diseases that would have occurred had not there been successful vaccines. So that's the first point I want to make about the importance of the whole issue of vaccinology and vaccines. Number two, when we talk about the development of vaccines, you can divide it into two general categories. At least I do that empirically in my own mind. And one is the improving on existing vaccines with new technologies, because we have some sensational vaccines. We have vaccines against diseases that we don't use the vaccine anymore. For example, smallpox with the eradication of it. We have vaccines against measles, hepatitis, polio, etc. However, there are some issues regarding the technologies in which we need to do better. Let me give you just two examples in the same disease. With influenza, the technology that we have been using has been a technology that's worked, but it's primitive. It doesn't belong in the 21st century, and that is growing the virus in eggs and then preparing it either in an inactivated form or in a live attenuated form. What we need to do is we need to get the 21st century technology of recombinant DNA, as well as even as basic as going from an egg-based to a cell-based. The other is that we need to improve the immune responses with adjuvants. And adjuvants are compounds which amplify not only the breadth but the depth of the immune response, how much of a response and how you go across cross-reacting. The reason I say that is that if you look at influenza in this country, each year 35,000 to 36,000 people die, and there are 200,000 excess hospitalizations. Forget about swine flu and pandemic flu. That's just seasonal flu like clockwork every year. 92% of those people who die, those 36,000, are elderly, greater than 65-year-old, and the other 8% are either infants, pregnant women, or people who have underlying conditions that compromise their immune system. So we need to make better vaccines for things that we are already successful. Then there's the whole area of vaccines for diseases that are important that we don't have vaccines for. And 3 of them are major killers throughout the world, particularly in the developing world. One is HIV. The other is TB. We have a vaccine, but it's not effective in the adult. It's partially effective in youngsters. It doesn't protect against pulmonary TB. And finally, malaria, which I'm sure you're going to hear a little bit more about. Point number 3. When I think of vaccinology, I think of classic vaccinology. versus unusual vaccinology. Classic vaccinology means that when we people who make, conceive of, or develop vaccines, we do it by mimicking nature. So if you look at the successful vaccines, smallpox, polio, measles, hepatitis, what the body has already told us, it is, as I call it, the proof of concept. It is proven that although 15 to 20% of the people die of smallpox and a few percent die, many get maimed by polio, people die and get very sick from measles. At the end of the day, when you are finished with the disease process, your immune system not only clears the virus from the body, but it really lends you to lifelong protection against subsequent challenges. So what it's saying is that the vast majority of times, your immune system has shown you that it's feasible and capable of blocking a microbe. So you don't have to be a genius as a vaccinologist to figure out how you're going to make the vaccine to act like the immune system. There are some technical hurdles that sometimes take years and years and years, but conceptually, you know you can do it. With a disease like HIV, that's a whole different story, because I get asked all the time, why don't you have a vaccine against HIV? And the reason is the body has not given us the proof of concept. For reasons we don't understand, there are really virtually no examples of an infected individual who has completely cleared the virus from the body. There are those who do better than others, but out of the 60 million people that are infected, there's not one documented case of somebody that has completely cleared the virus from the body, which means that we don't really understand yet what and if there is a correlative immunity that we have to get to. We have to do better than natural infection. I'm sure you're going to hear a bit about that from others, particularly Gary. So that's what I call special type of vaccinology. And I want to close with the fourth issue. And that really relates to what I said. H1N1, the swine flu that we're in the process, even as I speak today, of making a vaccine for, falls under a situation that's a little bit between the two. Because I can almost guarantee that we will have a vaccine against the H1N1 strain that is currently circulating. And the reason is that we have successfully made vaccines every single year for seasonal flu, plus or minus, depending upon the matching of the strain. So that falls under the arena of classical vaccinology. However, flu, like some other viruses, does something different. It has the ability to mutate and change. So the concern is, would you have a vaccine that perfectly matches the strain that is the currently threatening strain? So there's no doubt I can make a vaccine, or we can make a vaccine against what's circulating now. Hopefully that's the same one that's going to be circulating in the winter and the fall. And that's the reason why the new technology, which again I'm sure you're going to hear about, relates to making vaccines that are universal for a particular strain. In other words, an influenza vaccine that hits the parts of the virus that never changes. So even if it changes from one season to another, or it becomes a pandemic versus a seasonal, the vaccine will still work. So those are the four concepts, and I hope we get a chance to discuss them more. Joe?
Okay, thanks, Tony. Philip?
So Tony talked about the impact of vaccines on public health, but it's actually also impacted medicine and how it's practiced itself. And I'll give you a quick example. When I was in med school, I was taught that there were three classic pathogens of bacterial meningitis in healthy children. I'm a pediatrician. They're H. flu, pneumococcus, and meningococcus. And I took care of these children when I was in med school. By the time I finished my residency, 4 years later, H. flu was gone. That vaccine had been rolled out, and that disease entity had essentially disappeared. By the time I finished my infectious disease fellowship, the same thing had happened to pneumococcal meningitis. It was very rare, and it was a rarity on the floor, and people kind of gathered around to learn from it. And the same is going on now with meningococcal vaccines as well. And this is all based on one technology, on conjugate technology, where you link two proteins together to make them immunogenic and generate a vigorous immune response. Tony lumped it into two groups. I'm more in the weeds. I see 6 groups. I won't go through that and bore you. But each of those categories has spawned a series of vaccines to various diseases that have been rolled out and hold potential in the future. I won't go through all that because that would be tedious, but I thought it may be worthwhile to touch a bit on some challenges I see in developing new products, because that's what I do. I develop new vaccines. I think predicting the emergence of new pathogens is impossible. And even in the case of the H1N1 flu story, that almost was predictable. We know it happens every 40 years or so. And the U.S. government, took real leadership and set up a pandemic preparedness plan and funded both the public and private sector to put plans in place. So when it hit this spring, they were able to flip a switch, and all of a sudden you have capacity waiting and ready to make these vaccines, both on the R&D side and the manufacturing side, as well as some groundwork based on rolling out these vaccines. But we don't know what the next it is, whatever it is. So we don't know what's coming, so we have to be nimble in our approaches to developing vaccines when it comes forward. The other thing about vaccines is our target populations are healthy, largely children. And that means that the safety has to be taken very seriously and is paramount. And the risk-benefit ratio you have to think about is very, very different from other medicines. Since you're going into a healthy population with products that have some small risk, you have to be aware of that and make sure the benefit-risk ratio is appropriate before you move forward. Vaccines are getting more complex. We see it easily in increased valencies of some vaccines, like against influenza or pneumococcal vaccines. And we see it in the kinds of technologies we're having to put forward. We have generally not found good solutions to eukaryotic organisms, fungus, parasites. Tony talked about TB, malaria, and HIV, which has also remained elusive. And there are a number of bacterial organisms, staph and strep and E. coli, which we also have not been able to tackle appropriately. These new vaccines are coming. They're going to be complex. They're going to target multiple antigens. They're going to require new technologies, new adjuvant systems. They're going to be expensive to develop, manufacture, test. We have regulatory processes which have to be managed as we go forward. And this is all in face of a societal lack of valuation of vaccines in general. People value treatments very much. You can spend tens of thousands of dollars for treating a cancer, but you wouldn't want to spend the $400 for the vaccine to prevent the cancer in the first place. You can even spend that much money on a lifestyle like medicine, like Viagra, but people don't really grasp that they want to engage in preventive medicine in a real, appropriate, societal way. And that is a problem because it means that many potentially life-saving vaccines are being left behind. And I'll give you an example. Parvo B19 is a virus. In children, it causes fever and a rash. In people with sickle cell disease, it causes aplastic crisis. That means their bone marrow shuts down, and they often require blood transfusions. And in pregnant women, it causes hydrops, which often leads to abortion of the fetus. There's a vaccine which has been worked on and is likely to work. But the chances of it being moved forward and made available is almost nil. The cost to develop it, manufacture it, test it in these vulnerable populations is such an insurmountable requirement of resources that it will never be recouped on the other side, and no one is able to take that next step to bring that product forward.
Difficult. Okay, Gary, how are you going to fix that?
Well, I'm not sure I have those answers, but let me take a step back and then I'll come around to some of the issues at the end. To the theme that you laid out in this session of vaccine wars, I actually think it's helpful to think about vaccines from a strategic perspective. And an example that I thought might be useful or instructive here is when we think about the attack a few years ago that we all heard about on the USS Cole, a giant warship that essentially was taken out by a little powerboat. And in fact, when sort of military experts do their game playing, they're trying to figure out how to protect the battleships at sea, they actually have come to realize that one of the greatest threats to our warships is not another warship trying to fire on, not another airplane trying to take it. It's this swarming of a lot of little boats that could just come around a vessel unexpectedly and overwhelm it, and you'd have no way of essentially protecting against it. It's actually the same strategy behind pirates and the incidents we've seen in Somalia. Well, that's actually the problem we have when we're trying to protect ourselves against microbes. We're dealing with vast numbers of microbes, and it is a war. Just to give you an example, and it's a war in which the pathogens really have the upper hand. Take AIDS, as Tony mentioned a second ago. If you look at the number of viruses in a single person that's infected with HIV, there's well over 1 billion viruses. So in that one individual, there are probably as many viruses as there are people on this planet. And we're trying to protect that individual from that number of viruses. And the pathogens basically outnumber us. They multiply faster than we do. They will divide in a day or less than a day. They evolve faster than us. They're always changing their genetic makeup. And they develop resistance faster than we do for all the above reasons. So you might ask, well, how do we ever make a successful vaccine? We do it by relying on essentially the powers of the immune system that have evolved over our lifespans and over human existence, and our ability to recognize them and respond. But we're actually left with a lot of the nasty pathogens today that have figured out how to outsmart our immune systems. So I think that where we are now, particularly with the challenging vaccines, and the challenging ones would, of course, be AIDS, TB, and malaria. And I would also probably include flu on that list because it continues to evolve and evade our strategies, is that we really have to use what I call rational design, sort of the power of science, the power of biotechnology, and with our understanding of how these viruses infect us, and what parts of those viruses have to remain constant, we have to identify the Achilles heel on those viruses. And I'd say the two positive features, and I think two reasons to be optimistic that we can make some good progress in the coming years, is that some of these tools are very, very powerful, particularly in structural biology. We now can get atomic-level detail of the intricacies of those viruses, and we can pinpoint exact targets that need to be hit. It's as if your warship was being swarmed by these power boats, and you had a device that could turn off all the motors at once. You're safe. And that's what we're trying to do with a vaccine. And for those reasons, I actually think we now have some tremendous opportunities that we didn't have a few years ago. Tony mentioned universal flu. We know actually from some papers that have been in the literature, some recent, some a long time ago, that there is an Achilles heel on the spike of the influenza virus. And we even in the laboratory now can target that Achilles heel in such a way that we can generate antibodies that would neutralize most flu strains. In fact, in animals, we can generate antibodies against the earlier flu strains that will actually inactivate the current swine flu strain. So, for example, if we knew that vaccine had worked in people, we wouldn't have had to make a new swine flu vaccine. It would have already protected us. So that's our goal. Our goal is to take the latest in what we understand scientifically, to apply rational design, and to essentially use our intelligence to outsmart the viruses. The same is true in HIV, that there are these Achilles heels, conserved regions that can't change in order for the virus to be successful in infecting us. And the last thing I'd like to say is that we do need, I think about virus control and eradication in a more holistic way. We know that there are reservoirs of flu in animals. That's where the swine flu came. The pigs are a mixing bowl for these viruses, and we actually, although I agree with you that it's hard to predict and we don't know the rules, I think it's something we need to try to explore. How do viruses mix in animals? What makes them able to jump when they jump? And when can we actually control the virus infections in other species that then become threats to us? There's really only been one virus that's ever been eradicated. It's smallpox. And I think that that one example, though, is really a shining example of what we can do. In the 20th century, Despite the fact that we had a vaccine, there were still 300 million people that died from smallpox. It's an astounding number. Yet, when the vaccine was properly deployed and it got out there, we now literally have removed one disease that was a scourge on this earth for centuries, and it's gone. And I think that's what we've got to remember in this war, and I think that's how we're going to make progress moving forward.
Okay, Gary, thank you. Peter.
So I'd just like to talk for a couple of minutes about malaria. There's a lot in the lay press about malaria and malaria vaccines. People ask me, their children are going to Africa, can they have a vaccine to protect them? And the answer is malaria is a parasite. It's a complicated organism, 5,300 genes. It's probably been around for 100 million years. There are malarial species that will infect birds, reptiles, a variety of different mammals. And the life cycle is extremely complicated. For that reason, there's no simple vaccine. But if you ask someone from sub-Saharan Africa, a black taxi driver in Baltimore from Zambia, have you had malaria? They always laugh. Of course I've had malaria. Everybody's had malaria, many times. But of course it's a very difficult disease for children. But as the individuals grow up and have it repeatedly, it's less and less severe. There is a native partial immunity. So we have good reason to believe this organism is vulnerable to immune surveillance. The problem comes of developing a vaccine that prevents people from getting it altogether. And I'm speaking principally on the Falciparum malaria, the bad malaria in Africa, and protects children and protects women during pregnancy. And I won't go through the complicated life cycle, but basically this clever organism goes through basically 3 complete changes of costume when it goes from mosquitoes into the bloodstream, invades the liver, then into the red blood cells, then goes through a sexual phase of reproduction back into the mosquitoes. And if we could succeed at any one of these points, we might have a vaccine which would be useful. Now, I personally am not optimistic that in my lifetime we're going to have a perfect vaccine for malaria, but it's quite possible that some of the recent advances, and a lot of effort has been going into this, Gates Foundation amongst others, funding efforts, and it indicates that some of the vaccine trials have partial efficacy. The rate of individuals who develop malaria is somewhat reduced, small percentages, though maybe 30%, or it's less severe. So I think there's reason for optimism. But in terms of these other diseases we've talked about, I'm afraid malaria is probably we have the farthest to go. And I'm not optimistic we're going to have any big advances in the next couple of years. Sorry.
That's 2 downers we've ended on. I'm not sure I like the tone of this discussion. Okay, good. Well, all right, so we're off and running, and I'll take the liberty of asking the first couple questions, and then you're welcome to chime in. But I thought I'd start, Tony, by asking how you would address Filip's point about this economic problem, because the government has said, we'll buy all the H1N1 flu vaccine that you make. Don't worry about, the profit issue yet. But he's sketched the broader picture. So what do we do with that?
That is one of the real challenging problems, which is the fact that there is really not a great inducement to industry to make vaccines. First of all, for one reason or other, people are, and you said it very well, are less likely to pay for something that is going to prevent something than they are to pay for it after they get it. Because if they don't have it, they feel fine. So the idea of handing out X amount of dollars doesn't really strike them as being something that's cost effective from the standpoint of their health, even though we know from decades and decades of experience that it is in fact the most cost effective thing that we can do as a society. The other thing, I believe, is that the companies realize that the obstacles to their making and implementing a vaccine program are substantial, not the least important of which is the fact of the extraordinary attitude that we have in society now that vaccines are potentially more harmful than they are helpful. And there's a lot of spin about that. And the reason is that we don't see disease around us. So when you vaccinate your children for a variety of childhood vaccine-preventable diseases, you don't look around and see 10,000 kids dying from measles or a lot of congenital malformations from rubella in pregnant women, because it doesn't happen, because we vaccinate. So the idea of vaccine being something that's really going to save you is very, very difficult concept. So there's not a lot of momentum to get the companies to make the investment that it takes to get a vaccine that nobody wants to pay for and almost feels that if you're going to do it, it's going to be an entitlement. That's a very, very difficult situation, which is the reason why when we have situations in which, as a public health issue, the federal government realizes that we need it, the federal government gets in the process not only of partnering with industry, the way we did with building plants for some of the vaccine producers for influenza, but actually giving almost a guaranteed purchase that we'll buy it. So it's really a matter of the perception of the public and the lack of incentive for companies to put investment to make something that is not individually desirable, nor do people want to pay much for it.
Okay. And Filip, I wanted to ask you, so you're in charge of clinical development. Where are you putting your efforts? I mean, it's got to be partly a scientific decision and partly a business decision about deciding where to try to develop a vaccine. Where are you working now?
That's right. I mean, I think, we have to, we work for a private company, and we have shareholders, and we're responsible to them. At the same time, being a vaccine guy, I've been a vaccine guy since I was very, very young, I get to play very much a public health role in industry. It's a great place to be. If somebody wants to try it, you should really go for it. Because it allows me to develop life-saving products from get-go all the way to the end, where they can be commercialized and get gotten out to the public to save lives. So, we have an effort in influenza, and just including the H1N1 thing that's going on. We make the intranasal vaccine, and that's going very well, actually. We've managed to make quite a bit of it. We think we're on track to having product available by the end of August. So the thing that Tony talked about of the federal government paying for the manufacturing plants is right, but that alone won't solve it. You could build a plant, and then if you don't use it, then it goes fallow, and it's just wasted money. So what we really need to do is develop the demand for those vaccines year in, year out to keep those vaccine plants running and the R&D people smart and on edge. So when we do have another emergency, they're going to be ready to click on and deploy the vaccine. Just to get back to what I'm working on, my favorite projects are around RSV vaccines. RSV is a respiratory syncytial virus. It's a virus that causes pneumonia in young children. In fact, it caused my 2-year-old daughter to have a 7-day stay in Denver Children's Hospital. We were on vacation once, and she had an infection and had a pneumometastatic air leaked out into her chest, so we had to delay our vacation. Or just returning home. And this is a vaccine. This is an infection that causes severe illness in young children and has a checkered history from vaccine development. In the 60s, a vaccine was made and tested in the Washington area. And instead of protecting children who received the vaccine from getting ill, it made them sicker when they got infected with RSV. About 20% were hospitalized, and 2 children died in those studies. So that's an approach which didn't work. 40 years later, where are we? We're using another one of these classical vaccinology approaches, and that's to use a live attenuated vaccine and some host range restricted vaccines to start small infections in the nose, which will protect the children. And that's going very well. Right now we're down in infants, and the safety profile looks quite good with that.
Cool. And Gary, what I wanted to ask you is, I know you described the challenge. First of all, I wanted to clear up one thing. When you said that an individual who's infected with HIV would have 1 billion viruses, what you meant was 1 billion different viruses? I mean, they'd all be HIV, but they would be partially different? Or you mean there's 1 billion viral particles?
There are 1 billion viral particles. They're not 1 billion varieties of it, but there are a lot of varieties of it. Yeah.
So, thanks for. I just wanted to be clear about that. But I know you've been working on an HIV vaccine for a while. Can you give us a sense? I mean, this is. I mean, going back to Margaret Heckler's famous speech, we're a little past due the predicted date for a vaccine. That was the speech she gave back in the 80s when HIV wasn't known as that at that point, but the virus was determined to cause the disease. So where are we?
Well, it's been a long struggle, and I think the short answer is that we're probably optimistically no closer than 10 years, and pessimistically there are those who say we may never get there. To give you, an idea of what has happened in the field, there are two basic approaches that have been taken to the development of an AIDS vaccine. And they really stem from the two arms of the immune system that would respond to the virus. One is the cellular arm, T cells, and one is the humoral arm, and those are made by antibodies. And it's been a real challenge to develop what we call broadly neutralizing antibodies. And that early on was a holy grail. Well, a lot of effort was put into it, and we realized that it was not easy to solve. So that has kind of gone back into more of a research mode. That's where a lot of the information now is coming from structural biology. In the meantime, we said, okay, there's some good data that suggests that T cells can protect people from infection. So the T cell efforts have moved forward, and in fact had done reasonably well in early stage trials. The real slap in the face came 2 years ago when Merck was doing a study of their vaccine. It was a T-cell vaccine that used adenovirus, which is a cold virus, to deliver HIV genes. And what happened there was very similar to what you described with RSV, although maybe not as definitive. It looked like people who got the vaccine may have been at greater risk of getting infected, which is exactly the opposite of what you want. That put the whole field on its heels, to put it mildly. And I think that there's been a lot of examination of really what went on in that trial. And we're really not sure that is a viable way forward. Having said that, we're not sure that it isn't still viable to explore. This fall, in September, there will be another phase 3 trial that will be completed. And we're all anxiously awaiting the results of that trial. It's also largely a T-cell-based vaccine, a Sanofi-manufactured product with a VaxGen product. And we'll see. We'll see if there are clues. But we're in that exploratory phase where we're looking for clues from clinical trials. We're looking for clues from animal models. And we're going to move forward. The one reason to have some optimism, and I think this is a relatively new finding. Tony mentioned the fact that nobody who ever has been infected with AIDS has really cleared the virus. Unlike other viral infections, that's been a problem. There is one set of clues that's emerged in the last year, really, which is going to be important. And that is that when we look at large numbers of people infected with HIV, about 15 to 20% of them make those broadly neutralizing antibodies that we really want in a vaccine. So it does happen in nature. And what's happening now in the field is that we're madly scrambling to understand how that occurs and then to recreate that.
Mm-hmm. Okay, the final question, I'll just ask Peter, but maybe others want to chime in on this. And that goes to the point that Tony raised, and I guess Gary just referred to, that in every case, vaccines are used to give a leg up to the body's own immune system in the hopes that somehow the body will figure out what to do with this invading organism and get rid of it. Is there any other model? I mean, is there an idea that you could give an individual something more than his or her own immune system had to begin with, so that there would be, I don't, some superhuman ability, whether it's an immune response, for example, that a chimpanzee might be able to mount to a SIV, a simian immune deficiency virus, that you could give to a human that would enable it not to become infected. Has that ever been considered?
Are you steering that?
Yeah, sure. I don't look very clear.
The concept actually was out there, and that you can maybe fight infection with infection. And the Nobel Prize was once awarded for the use of malaria to treat neurosyphilis.
Not a good idea.
Thanks, Tony. Not a good idea. This was about
Volunteers
60 years ago. But I think the idea was that neurosyphilis was untreatable. And these profound fevers that were induced would, in fact, wipe out some of the spirochetes. So it wasn't illogical. It's just that the treatment is probably as bad as the disease. Maybe worse, I don't know. Would you rather die of neurosyphilis or of malaria? Neither sounds attractive. I think there are opportunities, though. The more we understand the immune system, and it's extraordinarily complicated. I don't understand it. So Tony Fauci must be the smartest person. I was told, Tony, is this true? You taught the entire immune system of Ronald Reagan? It's in fact dozens of different proteins that are working on each other. But can we in fact produce some of these and use them productively to block? And I think the possibility is there. Not in terms of malaria, but the more we know, sometimes the better our chances are.
Right. So it could become something someday completely different than the model.
That's the thing about science. Once somebody discovers something, it's like you're in a dark room, the lights are on. When Frederick Banting discovered this substance he isolated from dog pancreas could cause hyperglycemic children to become normal glycemic, it's like, whoa. We call that insulin. There was a point when there was no insulin, then there was insulin. So, I think discovery is really hard to predict. That's why I think we need to invest in this kind of work.
But I think there are some clues forward. I mean, now there's a big push, I think, in the community as a whole, working on different kinds of adjuvants, TLR agonists. And this is one way which we're able to actually achieve more with vaccines than we're able to achieve in nature alone.
Talk a little bit more about what an adjuvant is intended to do.
Want to do it?
Yeah. An adjuvant, if you take the body's response to a natural stimulus or to the stimulus that you give it, i.e. a vaccine, it makes a certain response. An adjuvant is a compound that magnifies the natural response to the antigen. If you just give somebody an adjuvant without the vaccine, you'll just give them a lot of inflammation and a lot of activation of the immune response in a nonspecific way. But if you give it together with the vaccine or whatever antigen you want to make an immune response does, it does one thing for sure. It increases the magnitude of the response to allow you to either use a lesser dose, it's called the dose-sparing effect, or it gives you a much higher level than you could get by the vaccine alone, namely when the vaccine level is too low and not effective. That's the primary reason why you want to give, or at least in Europe, they give influenza vaccine to the elderly together with an adjuvant because the elderly don't make a very good immune response against influenza, and you could bring their immune response up higher. The third advantage, potential advantage of an adjuvant is that not only does it make the immune response higher, it makes it broader. By broader, we mean if you take an influenza virus and you give it as a vaccine to someone, they may make an immune response against a certain components. Then when the virus drifts away a little or changes, your vaccine may not adequately address the changing virus. If you give it with an antigen, the breadth of the response increases. So it's the depth or the magnitude of the response and the breadth of the response. So it's sort of like an adding, jazzing up of your immune system against the specific thing that you want it to be making a response against.
And if I can just, I'll go to questions in one second, but there's one other question I want to ask. Is there a way to measure how activated a given individual's immune system is? In other words, can you start with one measurement before the vaccine and another one afterwards and say, oh, we see how much activation there is? There is a way.
There is, but we often don't know if we're measuring the right thing.
Right, exactly.
So it's a bit of a black box, and we're shining our tools, our flashlights in there, and we see something, and we say, oh, yes, it increased it. But we don't know if that's what we were trying to actually achieve.
All right. Yes, in the back.
Well, just to follow up on the question about adjuvants, we have never had a flu vaccine which
Yeah, we're doing clinical trials well, we aren't doing them. We're going to start them in August, and we're going to be looking at the effect of certain adjuvants together with H1N1. Since we don't have experience with adjuvants in young individuals, the Europeans have it with elderly individuals, it is unlikely that an adjuvant will be used with H1N1 without what we call an emergency use authorization, because it would take a lot of data that would take too long to be ready for the flu season to give it. So if the situation gets serious enough that you really do have to use an adjuvant, like the disease changes, it becomes much virulent, we don't have enough vaccine, that we would use an adjuvant, that's the circumstance that we would have to do that. But clinical trials are being done. The Europeans have it licensed for influenza.
Yeah, go ahead.
First, who makes the emergency use authorization?
The emergency use authorization is a authority that is given by law to the Secretary of Health and Human Services, who imparts upon the FDA, which reports to HHS, the ability to use a vaccine or a drug before it is formally licensed.
Can I ask, you mentioned earlier on the social issues we now have increasingly in this country with vaccines. From the point of science, what can be done to deal with that, especially thinking ahead to if you need to get a big program with an H1N1 vaccine, that could be a real test of this. I mean, is there any sense as to whether people would go along with it?
Well, that's a very good point. If there is, and that really leads to the balance between the risk of a disease versus the risk of a vaccine. If there is no disease, if somehow magically H1N1 disappears and doesn't come back in the fall, that's not going to happen because it's already rampaging through summer camps and army installations and military installations. But let's say magically it disappears and all of a sudden September and October comes and says, all right, we want everybody to get vaccinated. You're going to have very poor accrual. But if you look in the southern hemisphere and there's a lot of disease in the schools and the school children are getting sick, and as they come back to school in September, schools are closed and classrooms are decimated, you don't have any problem with getting people taking the vaccine. We will be criticized for not having enough vaccine in time, as opposed to giving people something that's going to hurt them.
Yeah, over there.
A question about the cholera vaccine. There is one, but it's not very effective. Cholera is a disease that really does affect most of the people elsewhere in the United States. I wonder if anything is being done to improve that vaccine or make it more widely available.
Well, it's an enteric disease, and we have different classes of antibodies in our internal compartments or in our secreted fluids. And antibodies that react with intestinal bacterial infections are probably much less effective. I think that the vaccine has some efficacy, but the prevention is pure drinking water, of course. And if we could provide pure drinking water, then the vaccines will probably not be particularly useful because the infection rate will be so low. In terms of making it more effective, I don't know.
Well, you know, there was a real problem with cholera years ago, which was one of the real examples of why you better do a good clinical trial before you license a vaccine, because the cholera vaccine was developed without very good scientific evidence that it worked. It was licensed, and then about 10 years later, they found out that it was completely useless and didn't work. So people are working on a good cholera vaccine now.
Yeah.
So with the experience of the adjuvanted seasonal flu vaccine in Europe, when do you think that product would come to the United States?
Well, that's a good question. I can't predict that, but the FDA will have to examine very, very carefully the vaccine data that comes from Europe, mostly in Italy. For example, Novartis has vaccinated over 10 million people in Italy who are greater than 65 years old with seasonal influenza. They have apparently a pretty good safety record. They haven't yet, or at least in the process of submitting it to the FDA, for the FDA to actually examine the data to make that determination. Also, we will get data if we use an adjuvant, and that's a big if. we use an adjuvant in adults. It's extremely unlikely. This is not a policy statement, underlined, but it is extremely unlikely that we're going to use adjuvants in young children in school. But if the Europeans and others use adjuvants, which it looks like they very well might, in their vaccines for young adults in Europe during the coming flu season, then there'll be a lot of data that the FDA can examine, in addition to our clinical trials.
And the U.S. government is preparing for this. They've already bought it. So
We have
it's on
120
hand.
million doses of adjuvant that we've already bought.
Is that the same product that Novartis uses?
There are two types of adjuvants that are out there. One is ASO3, which is a GlaxoSmithKline that's being used by Sanofi in a mix and match, and the other one is MF59, which is a Novartis one.
Maybe I'll ask another question just because I realized I'm curious about it. This goes scary. I had understood that when the SARS outbreak occurred, you took some of your lab's efforts and started looking at that. What happens when you go down a path and then, as Tony says, magically the problem goes away, but you don't want to let it go in case it comes back? I mean, how far did you get with SARS?
That's a great question, Joe, and an important one in terms of how we respond. We actually made a vaccine, a DNA vaccine, that was fully protective in the one animal model.
Can you explain what a DNA vaccine is? Just in case
a DNA vaccine is simply a piece of DNA that's grown in bacteria, and we just purify that DNA. It's been engineered in such a way that it will make one or more of the proteins that comes from the virus. So it can't make the virus as a whole. It makes that one gene that normally the virus would make, and your body sees it, responds to it, generates antibodies and T-cell responses. And it's very safe. It's probably, it's yet to be shown effective in humans because it's not as potent as some of the other delivery methods. But there's a lot of room to improve that technology. In any case, we developed a vaccine that went into phase 1 trials. Others have developed vaccines that have gone forward. The news was all good. We actually immunized people. We generated neutralizing antibodies in people. Others made other vaccines that did the same thing. And then we ran into the problem that I call we were all dressed up with no place to go.
You couldn't find a date.
Yeah.
Well, I'm sure we could loose it on the world, and you'd have people to test it on if you want.
But it's an important issue because if you have a vaccine and you want to be prepared, and yet you're not in a position to do a trial to prove efficacy, then you're kind of stuck. Now, the FDA has created a rule to help with this situation. It's called the animal rule. And what
Two-animal rule.
Well, actually, that's a matter of debate. It's at least one.
It's at least one.
Preferably two. And what they ask is that you test the vaccine in an animal model that is very similar to what you see in humans. And then you define an immune correlate based on your study of that immune response in the animal. You then test the same vaccine in people, but of course you can't challenge the people. So you just go on and say, okay, I generated that same immune response in a person, so it ought to work. Mm-hmm. The problem in SARS was that we had animal models of SARS infection, but they weren't similar to what we saw in humans. Mm-hmm. And so there literally, for that vaccine, is no way right now to get to licensure unless we can develop models. Now, the latter could probably be done, but the disease is pretty much gone. And what company would want to make that vaccine when the disease is not out there?
But I have a concept now where Tony could fund a new program called America's Top Animal Models.
Right.
We could use that. Sorry, somebody knows I make those jokes. Yes, go ahead. Sorry. Yeah. Sorry. I looked past you, but I didn't realize your hand was. Never mind.
It's perception, depth
right,
perception.
It's depth perception.
I
Right.
guess I have a question for Tony. And Tony, you had said that the influenza found an acute apparent Achilles heel in the influenza virus, that the portion of it doesn't change.
Right.
It appears not to change.
Right.
So I have a couple of questions related to that. First question is why would that be the case? And in fact, is that a portion that's related to transmissibility? And then the other question, which is actually related to how do you define an influenza virus? And as we go through successive series of mutations, generations, when does it cease to be what you would call an influenza virus?
There's no mutation that would happen that would make it another virus that you wouldn't consider, because it would just mutate to being non-replicative. But it would still, so it isn't as if you're going to mutate so much that all of a sudden you've gone from an influenza virus to a coronavirus, because the genetic makeup is so different that you may get an influenza virus that mutates enough, it's drug resistant, it's this, but then it's just a completely non-fit. We use the word fit. It's a non-fit virus. It essentially doesn't replicate. But the point that you were asking about, there are several, your first question, there are several components of the influenza that are really constant. One of them is the M protein. The other one is, one of them is the one that Gary mentioned, the stalk of the hemagglutinin. So the hemagglutinin is like a bulb with a stalk. And this thing here, it doesn't get recognized very well. And when antibodies come, they tend to keep making it against the hemagglutinin. And the hemagglutinin is structurally made so that it almost as if you don't see this. So what Gary and others have done have been working on the stalk of this, which gets to your question of the transmissibility, because HA has everything, the hemagglutinin has everything to do with being transmitted. So even though the part that's the bulb is the one that changes, this doesn't change. So what Gary and others are working on is trying to make an antibody against that constant stalk.
Transmission, then it just dies within wherever it is.
Well, it doesn't die. It can't be transmitted effectively.
What it does is we actually think that structure that doesn't change is actually functionally required for the protein to attach and then enter. And what it does two things. There are actually three of these. And we think that region is critical in holding the three together. So anything that the virus does to try to change it, that falls apart, and you now have a non-functional virus. Right. The other thing is if you do generate an antibody to that region, essentially what it does is it just freezes the hemagglutinin in place. It just can't move, paralyzed. So for that reason, it is attractive, but it's probably not as potent as the kind that Tony was just talking to the head region. Right. And so it still has to be explored, and we still have to assure ourselves that we can accomplish that in people. But it's certainly an exciting target. And there are some monoclonal antibodies that have been directed to it that have very impressive broad neutralization. So there's a proof of concept.
That was just discovered this year, by the way, that area of the stalk, just literally this year.
There's a lot of public furor about vaccines with misinformation. I wonder if the other panelists would care to comment on what is the scientific evidence linking or not linking the preservatives with the appearance of autism? Yeah. Is there any scientific evidence? And the second thing, and then is, of course, the human cervical cancer virus. What is the scientific information you'd like the public to know?
Well, having testified at about 185 congressional committees on this, just joking. That's a hyperbole.
Advise and extend your remarks later.
Yeah. There is really no evidence whatsoever that the amount of thimerosal that is in the ethyl mercury that's in the preservative of a multi-vial dose has anything whatsoever to do with autism. The data have been examined every which way but Sunday by everybody.
So this is another conspiracy theory?
Well, I wouldn't say conspiracy, because you've got to be careful that the people who are doing that have suffered a lot because they have had tragedies in their family. So I don't think they're deliberately doing anything in a conspiratorial way, but there needs to be a reason why they have, a situation where there's a devastating illness in the family. And the problem with vaccines, and this is maybe more generic to your question, Peter, the issue why vaccines have such difficulty and always getting blamed for virtually everything and anything that happens is that you give a cluster of vaccines to children during a certain period from shortly after birth until their X number of years, And when children start manifesting diseases, everything from neurological components like autism or a variety of other diseases, it occurs during that period. And people say, well, my child was vaccinated 3 weeks ago, and then I found out that they had juvenile rheumatoid arthritis. Or my child was vaccinated 2 weeks ago, and I found out that they had X disease. And when you're looking, and you're a frustrated parent, and you're looking for, why is this happening to me? It was the vaccine. I mean, it's almost like saying that you could say that you go into McDonald's, and 1% of the billion people who come out of McDonald's drop dead on the sidewalk for the heart attack. Therefore, McDonald's causes heart attacks. Maybe they do. They do. That's right. It doesn't happen that rapidly, though.
The better example I heard was lotteries cause deaths.
Right.
Because if you calculate the number of people who die on the driving to the store to buy their lottery ticket.
Right.
Anyway, but your other question, what about the other question about
cancer.
cervical cancer?
Well, go ahead, Gary.
Well, actually, before I say something about it, I just want to amplify on one point, which is that I do think it's really incumbent on us, and this is where it really is a challenge in vaccines, because we deal with many relatively rare diseases that are hard to study. So, for example, with autism, I actually think we really need to figure out what causes autism.
Right.
And I've spoken to parents whose kids have had vaccines and had autism, and it's very difficult. And you almost can't reason because it's such a personal, passionate issue. But what I say to them is, look, if the vaccine causes autism, I want to know. And on the other hand, if the vaccine doesn't cause autism, we need to find out, because we need to know how to treat your child and how to prevent this from happening in the future. So I think we absolutely have to take any complication of a vaccine seriously, try to understand its true nature, and it's tough, but we just have to be transparent and ask those tough scientific questions until we get to the other side.
Again, just if I could, before we get to the HPV, and maybe this is, I think it is important for the audience to understand the point that Gary was making about when we test the vaccine, we document everything that happens following the vaccine, even though by common sense you know that it had nothing to do with the vaccine. Like someone gets a vaccine today and 2 weeks later they get diagnosed with a brain tumor that had to be there for a year before you notice it. That gets recorded. So it isn't as if there's a cavalier, careless way of looking at every possible thing. And then what you do to determine whether or not it's related to the vaccine is that you look at the occurrence of that in the natural population. So if somebody gets vaccinated with a typhoid vaccine or a yellow fever vaccine because they're going to go travel to the developing world, and they walk out of the doctor's office, if we were doing the clinical trial, and you got a heart attack, that would be recorded as a potential adverse event. But then what you do is you go to the broad picture of how many people who walk into a doctor's office and walk out get heart attacks, and you find out that that associated with that injection was no greater than what's going on in the general population. That's how you determine the difference between something that's an event that's completely unrelated versus something that's causally related. And because of that, we know that a lot of the claims that we were talking about that people make that are associated with the vaccine is no different than the occurrence in the normal population of people who don't even get vaccinated.
And I just want to close the loop on Peter's other question about the human papillomavirus vaccine.
There is no question that there are data right now that the data are actually accumulating very, very well. First, it was 11 to 12 years old, and you had to vaccinate your child, your young daughter, early, because otherwise they felt that if you vaccinated after they got exposed to HPV by sexual contact, that the vaccine wouldn't be effective. Well, now they're showing that in 13 to 26, it definitely decreases the incidence of HPV. And now there's even some studies that are showing beyond that. So the benefit of the vaccine is starting as years go by and you get more data to be shown to be a real benefit. The benefit is in preventing the precancerous lesions associated with HPV. There has been not enough time yet to definitively show that the benefit is a decrease in cervical cancer, because the vaccine has only been used for a few years. It will take studies that go out 10, 20 years to show that people who were vaccinated and protected from certain strains of HPV actually not only had a decrease in the precancerous lesion, they also had a decrease in the actual cancer itself. Now, early on, there was some same claims that there was some neurological complications, seizures or what have you, that occurred with the injection of HPV. Now that there are essentially millions of cases of data showing that there are no serious complications associated with it. But early on, it was the same thing that we dealt with in kind of the mentality that a child somehow went into the office, got the HPV vaccine, and had seizures a few hours later. So there was a scare, or there are neurological difficulties associated. Then when you did an additional few million kids, you found out that there really wasn't anything associated with it.
Plus the way that vaccine is made, it's extremely safe.
Right.
It's a reconstituted pure protein vaccine,
Right.
so people can't get an infection from it.
Right.
It's a nice vaccine.
Yeah, go ahead.
I'm wondering, is it necessary to, you read all different things, revaccinate people? And, you vaccinate when they're young, and then people think they're good for life. Is it important to?
It depends on the vaccine. Tetanus, you need every 10 years.
Measles, probably one dose.
Yeah.
And there's some of them we don't even know the answer. Smallpox, we don't know if you really need to be reboosted. Polio, it is recommended that you get a booster as an adult.
Right. Yeah, but don't get nervous. You didn't get your booster. The reason is that we almost eradicated polio. So if you almost eradicated polio, then it is unlikely that you would need to have boosters if you got vaccinated before. But now there may be some rare cases of it, so adults may find out that they're vulnerable.
Yeah, and I should clarify that. You would get it only if you were going to go to an area where you were going to likely to be exposed.
Hepatitis?
Well, hepatitis, if you get 3 shots, and after the last shot, you look at your antibody level. If it's adequate, you're done. You're good.
Go ahead.
B.
B.
Okay.
Good question. No, we're hoping we'll get it.
Lots of work going on
Yeah.
that.
Go ahead. Well, I was going to ask this.
I actually got nervous because you mentioned smallpox. And my son, when he was little, he got a convulsion 2 days after getting the smallpox vaccine. So, yeah, naturally, I made the connection that it had to do with the smallpox vaccine.
Right.
Well, I saw the Mets win the World Series right after I got vaccinated. So I was pretty convinced those were related. Yeah, go ahead.
With the childhood vaccines, I have a daughter who's got a friend who says that, not that anybody has come up with autism, but now we cluster more of the childhood vaccines, because I think it has something to do with payments. We used to go every month, to have your child vaccine, and now it's been cut down to everything gets bammed in, within 3 visits within the first year. So now, have you tested that?
Yeah.
That
We are now, we just started in response to the same kind of natural concerns that may not really be justified in the scientific sense, but certainly justified in the concern sense, that there's reason to be concerned. We're looking at clustering versus individualing and see if there's any difference there. But you have to remember that the amount of antigen that is put into a vaccine, even if you take 10 vaccines or 11 vaccines, is minusculely less than the number of antigens that your body gets exposed to at any given time. So it isn't as if you're challenging your body to something that doesn't happen to it constantly anyway.
There's also...
I'm checking on it because these young people, there's so much spinning in their heads.
Oh, that's what we were talking about earlier.
I mean,
Right?
it's just constantly spinning.
Right.
The rotavirus, too, is a new vaccine, which I think is really excellent. And our grandson did get our RSV.
RSV, yeah.
Yeah, and now we were told that can
That's right.
Because once you're, exposed to that, you have to be careful.
Yep, we all get it every year.
Yeah, go ahead.
RSV.
I'm so impressed with what all of you are doing in the laboratory, and yet one of the recurring concerns you express is in consumer or societal confidence and attitudes towards vaccine. How much are you investing in learning how to build consumer confidence in vaccine? I know in Great Britain, for example, with the resistance to MMR, they found in the National Health Service that if they gave people simply the option of doing what this woman has recently mentioned, not lumping all the vaccines together and taking their kid every month to get the old-fashioned series of vaccine, that resistance to MMR decreased markedly. How much of NIH's time is spent not on laboratory vaccine fixes, but on fixes to poor humans and their prevalence?
There is an effort going on, and there has been, to try to educate the public about the use of vaccines. I don't know that it so much has originated from NIH.
No, it's a CDC issue.
It's CDC and also some of the societies, like the National Foundation for Infectious Diseases. They have taken some very proactive outreach efforts, both in terms of educating, trying to get TV programs on where they show what happens if somebody didn't get their vaccine. So that's happening, but it's really not something that we're taking primary responsibility for at NIH.
There are also disease-specific advocacy groups, usually founded by families who have been struck by disease that was vaccine preventable, and those are a powerful public force, to get word out.
Peter, I want to ask. I'm gonna take one more question from the floor, but I want to ask one other question just apropos of something you said at the start about the malaria vaccine being a long ways off. But isn't there a phase 3, a large trial that's about to commence?
Yes. There's.
So there are products.
Sure. A lot of work is going into this. Right. It's very important, if this can be successful, that we achieve it rapidly. So this is a complex vaccine with an antigen from the pre-erythrocytic phase of the life cycle. So the idea is the antibodies will prevent the sporozoite parasites from passing into the body and getting into the liver. And in fact, there were 3 trials that I'm aware of. 2 showed marginal effects, but then there was a very important study in children in Mozambique, which showed a partial efficacy, maybe I think a 30 or so % reduction in the rate of infections, meaning still it's not driven down to 0. And then the severity. I think based on that now, with new adjuvants, and I'm not up in the very latest, and that maybe Tony or Carrie is, they're going to try harder to use that as a sort of an entry pathway, sort of the lead. But again, it's a complex thing, and when you raise antibodies to complex parasitic or mammalian antigens, it's difficult with viruses, but it's really difficult. There's an illustration I just briefly like to share, I think. How many women here know their Rh blood type, Rh positive or negative? Many. My mother never went to college. She knows she's Rhd positive because of the importance in maternal fetal immunity. But despite the advanced state of science now, we can't recreate the Rh antigen and boost people so that this is to protect them. It's something that we can't do in the laboratory. It's so complicated. And so I think, knowing that, we should try harder. But we have to be respectful. Nature isn't always so easy.
Fred, why don't I let you ask the last question, if you still remember what it was.
Well, give it a shot. I was recently in Cambodia, and one of the things that we kept hearing about is the specter of H5N1 and H1N1 combining.
Right.
The whole superbug scenario. And I wonder whether that is an enabler in some ways for vaccine programs ultimately, because that's the kind of thing that just scares a lot of people.
Yeah.
You might have to address the possibility of it as well.
The only problem, it would certainly scare people, and that's what we talk about. What we're saying is that viruses, particularly influenzas, have 8 genes, and when you mix up an infection with one influenza with another in the same person, that they could switch genes. So the H5N1, they call that reassortment. The H5N1 doesn't transmit very well, but when you get infected with it, it's very deadly. There have been only 450 infections and about 280 deaths. H1N1 transmits extremely well. Test assessed, it went from a couple of cases in California and Mexico to 160 countries now in just a few months. But it is a relatively mild infection. So the concern is, as H1N1 works its way around to the regions of the world, Southeast Asia and places like that, where there is H5N1, is there a possibility of that recombining and coming out with a hybrid that has all the transmissibility of the H1N1 and all the deadliness of the H5N1? The answer is that is possible. The chances of that happening, you can't really say, but is likely low because it is not really recombined very much with the regular H1N1 that's going around in society. So you can never say never and never say always on this, but the chances of that happening are low. Yeah.
Well, we've scared everybody in the room. That's good.
That's good.
All right. I hope you'll join me in thanking the panel, and we'll draw this to a close.